So very good morning to everyone. How are all of you children? Good morning, good morning, good morning. Am I audible, visible, everything all right? Yeah. Yeah, I'm fine. Good morning, good morning, good morning, good morning. So let's start the today agenda. Right? So we are in between to discuss the difference between prokaryotic and eukaryotic cell. We just started it. I'm repeating it also, don't worry. I'm starting from a scratch. So prokaryotic cell, and here it is eukaryotic cell. If you talk about pro, name is saying about it. Pro means before. And karyotic, the word is karyon, means nucleus. So what the meaning is saying? These are the cell evolve before the evolution of nucleus. Got the idea? Before the evolution of nucleus. So what we can say here, in prokaryote, true nucleus is absent. And if you talk about U means true, karyon. So here what is present? True nucleus is present. True nucleus is present in eukaryotic cell. But in prokaryotic cell, true nucleus is absent. Now a question arises, if true nucleus is absent, then what is present? You will ask a question, na? Who will do the duty of nucleus? So what we are going to talk, in place of true nucleus, nucleoid is present in prokaryote. Nucleoid is present. Got the idea? Nucleoid is present. True nucleus is not present. Nucleoid is present. These nucleoid, it is also called genophore. These are the different name of the nucleoid. It is also called incipient nucleus. Or it is also called double-stranded circular naked DNA. Got the idea? So these are present in place of nucleus in a prokaryotic cell. I told about it in the last class as well. Got the idea? So I am drawing the diagram here. You just assume this is a prokaryotic cell. Assume here you will got a basic difference between nucleoid and nucleus. This is a assume prokaryotic cell. So in prokaryotic cell, I am just drawing the structure of a nucleoid. So this is the nucleoid. What you are finding? Single Single means one. Circular. Double stranded. Naked. DNA. Got the idea? Or this is called what? Nucleoid. Got the idea? Clarity came in the mind? In eukaryotic cell, what is present? This is a cell. Eukaryotic cell, assume, or you make a, suppose that this is an animal eukaryotic cell, and in the cell, look here, it is a double membrane bounded, nuclear membrane is there, fluid filled matrix, nucleoplasm is there, dense material, nucleolus is there, and in this one, network like a structure chromatin here, and this structure is called nucleus. So this is bounded by membrane. It is nuclear membrane. In short, I am writing. This is fluid called nucleoplasm. It is nucleolus. And this is what? Chromatin network. In this chromatin network, how is there? DNA is there plus protein is there. DNA, RNA plus protein is there. If we talk about composition of the chromatin network, so I hope you are able to understand the difference between nucleoid and nucleus. Can I expect? Are you comfortable now? Eukaryotic cell have a well-defined nucleus. What is the character of a well-defined nucleus? A well-defined nucleus have, see, inside the cell it is surrounded by two membrane, that is called nuclear membrane. Inside this, I am just drawing a nucleus only. This is the two membrane. It is called nuclear membrane. Inside of this fluid is there. This is called nucleoplasm. Then there is a dense material is there called nucleolus. And later on, you will find here, there is extended network like a structure is there, that is called chromatin network. I have given the labeling of everyone. Got the idea? So if such kind of a structure is there, it is called true nucleus. Means true nucleus have its own boundary. You see, here it is naked DNA, it is present in cytoplasm. So nucleoid, where it is present? Present in cytoplasm. No special compartment is there for the DNA in the prokaryotic cell. But here for the chromatin in which DNA, RNA and protein is there is a special compartment is there. So this is nucleus and this is nucleoid. This is only DNA and it is a nuclear membrane. It is nuclear membrane. It is a boundary. Two membrane structure are there. Got the idea? Is it clear in the mind? It came. So are you able to differentiate between what is the difference between nucleoid and nucleus? So one difference between prokaryote and eukaryote cell is over. In prokaryotic cell, nucleoid is there. Got the idea? In eukaryotic cell, nucleus is there. Done. Now I am coming on. One of the student asking, in prokaryotic cell membrane is there. Cell membrane is present in every cell, either prokaryotic or eukaryotic cell. I am coming on. Just give your attention. Now I am writing some more difference between prokaryotic and eukaryotic cell. And why do not that use that same slide? Wait a minute. I am going to talk some more difference between prokaryotic and eukaryotic cell. So in prokaryotic cell, what you will find, In prokaryotic cell, you will find all the membrane bounded means who are surrounded by membrane cell organelles are absent, and in eukaryotic cell present. All the membrane bounded cell organelle are present in eukaryotic cell and it is absent in eukaryotic cell. Means who is absent? Who is membrane bounded organelle? Example, mitochondria, chloroplast. These are bounded by double membrane. It will help you, double membrane bounded. Endoplasmic reticulum, Golgi complex, lysosome, vacuole. These all are what? Single membrane bounded. Single membrane bounded. These all are absent in which one? Prokaryotic cell. No, none of these cell organelle is present in which one? Prokaryotic cell. So here what we can say? Membrane bounded organelles are present. Membrane bounded organelles are present in which one? Eukaryotic cell. Yeah, beautiful question. One of the student asked, if mitochondria is absent, sir, then how the cell will get the energy? Because mitochondria is called powerhouse of the cell. Got the idea? Two questions came based on which one? Mitochondria. Two of the students asked. Got the idea? How they perform photosynthesis, how they perform respiration. Got the idea? Right? So all the job of the mitochondria plastid are performed by plasma membrane. In place of mitochondria, mesosome is there in prokaryotic cell. I will tell you what is mesosome. When I will teach you about the prokaryotic cell in detail, I will say function of mitochondria performed by mesosome. Got the idea? And function of chloroplast in photosynthetic bacteria is performed by chromatophore. Got the idea? Is it clear in the mind? So glycolysis occur in cytoplasm. So much curiosity is there. I am just happy to see it. So many curiosity are there in the students. Very good. Keep it up. Keep it up. Right? So you will got the answer of your every question. Don't worry. Got the idea? Yeah? So have a look now. So you got the idea? I will tell you about whose function performed by whom. Wait for that. Meanwhile, I am saying only the difference between prokaryotic and eukaryotic cell. I will also teach you in detail about prokaryotic cell. Got the idea? So these are the things I wrote. Mitochondria, chloroplast, endoplasmic reticulum, Golgi complex, lysosome, vacuole. These all are absent in prokaryotic cell. So membrane bounded organelles are present. So these are the organelles which is present in. I also have given you information, small information. That is what? Which cell organelle are double membrane bounded? If someone going to ask which cell organelle are double membrane bounded? Mitochondria, chloroplast, and which one single membrane bounded? Endoplasmic reticulum, Golgi complex, lysosome, and vacuole. Got the idea? Is it clear? Done? Right? I will answer everything. Don't worry. Don't worry. Don't worry. Have a look now. Now give your attention. In prokaryotic cell, already upside I wrote prokaryotic cell, so I am writing 70S type of ribosome is present. So what is 70S? I will explain when I will teach you the ribosome in detail. 70S type of ribosome is there. Here, both 70 and 80S are there. 80S ribosome present in cytoplasm and 70S ribosome present in mitochondria and chloroplast of eukaryotic cell. Means overall inside the cell of the eukaryote, which ribosome is there? Both 70 and 80S. What is 70S and 80S? I will teach you later, when I will tell you about what? Ribosome. Got the idea? So you just keep one thing in mind. Your question is what? All the question get satisfied when you will find the chapter it is going to close. We just started. We are just talking about the difference between. I know your curiosity. You know, you will ask what is 70S, what is 80S, what ribosome is doing. So I am just telling meanwhile that ribosome is a non-membrane bounded cell organelle. And that is the reason it is present in prokaryotic cell also. Non-membrane bounded cell organelle. Your every question is going to justify it, satisfy it, but wait for that. Have some patience, because we will discuss this particular thing on the right time. Got the idea? So ribosome is a non-membrane bounded cell organelle. So 70S type of ribosome present in prokaryotic cell. If someone asking a basic difference, so you can say 70S in prokaryotic cell, 80S in eukaryotic cell. But sometime paper setter becomes frame this question very smartly. Which type of ribosome present in eukaryotic cell? They can give a question. Always answer of a question depend upon the options. So they can give a question. I am asking a question here. ATS. Wait. Which type of RNA? Sorry, which type of ribosome is present in eukaryotic cell? Option I am giving. First option 70S. Second option 80S. Third option 90S. Fourth option 100S. What the answer will you give? Eukaryotic cell. You will answer one or you will answer two? So correct answer is ATS here. Because we have no any option to choose 70S. Now, same question I am framing and I am giving the different option. First option, 70S. Second option, 80S. Third option, 90S. Fourth option, both 1 and 2. What the answer you will give here? Here we will answer both 1 and 2. So see na, question is same. Question is entirely same or not? Tell me. Exactly same question. I not make any alteration in question. I simply change the option. In one question, answer become only ATS. Got the idea? Because generally what we find, what is the difference between ribosome of prokaryotic and eukaryotic cells? So we are saying 70S present in prokaryote, ATS present in eukaryote. We said like this or not? Are you getting my point or not? Because here we have given both as an option. We have an option to choose 70S also, and we know inside the mitochondria, inside the chloroplast of eukaryotic cell, who is also present? 70S ribosome is also present. So here I opted for fourth option. Got the idea? So questions is not powerful thing. Who is powerful thing? Options. Same question you can get many answer based on the options. Got the idea? One student asking, why should we write ATS if 70S is also present? See, major differences. Normally we are finding prokaryote difference 70S, eukaryote what? ATS. Got the idea? And we have no any option to choose. So what is the specific quality of the eukaryote? Who is differentiating eukaryote from prokaryote? Here ATS is there. So major differences created by ATS, na? How you can create a difference between prokaryote and eukaryote based on ribosome? So you can say prokaryote have 70S, eukaryote have 80S. But options is there 70S also, and you know that this 70S also present in the mitochondria of and chloroplast of eukaryotic cell. So we have opted here both. That's the reason I am teaching you, na? That's the reason I am just trying to give you this lesson. Ki same question, answer may vary, it's depend upon the option. One student asking, daily morning classes, sir, yeah. Is it not suitable for you? Morning class you are finding difficult? Yeah, same. If 70S and ATS both are given, we have to choose ATS because it is differentiating eukaryote from 70S. If both are given, we can choose both. That's the reason I framed this question. Lysosome is a single membranal, right? Na? Lysosome also a single membrane bounded organelle. Ribosome is not a membrane bounded organelle, and it is present in both prokaryote and eukaryotic cell. Now I am going to tell you something more about the prokaryotic and eukaryotic cell. Simply keep in mind, otherwise from every point you can arise a question. And it is not possible to fix every question at the given time. You also know it. So I am asking here, cytoskeleton is absent. So you will ask, what is cytoskeleton? I will tell it later. Cytoskeleton is absent in prokaryotic cell. I will tell later about cytoskeleton. What is cytoskeleton? How many type of cytoskeleton? Simply you keep in mind, cytoskeleton is something sir wrote. It is absent in prokaryotic cell. Later on he will tell much detail about what? Cytoskeleton. So cytoskeleton is present. Cytoskeleton is present. Now I am also writing one more line. Definitely I know you have question based on it. And that question is also relevant. But I will answer all these questions at the right time, not right now. Cytoplasmic streaming absent. Another question came now, what is cytoplasmic streaming? I will say, don't worry. Cytoplasmic streaming absent in prokaryotic cell, and this cytoplasmic streaming is present in eukaryotic cell. Got the idea? Another difference is I am writing in prokaryotic cell, no compartmentalization. Here, cell divided into many compartments. I am changing the color. This line I am explaining. Dividing right now. into many compartments. See, why I am telling prokaryotic cell no compartmentalization is there, because it is the cell membrane. Below the cell membrane, no any organelle are there who have membrane. Got the idea? So, if you talk about eukaryotic cell, it is mitochondria, it is plastid. Are you getting the idea? It is endoplasmic reticulum. So, every cell organelle making a compartment. So because membrane bounded cell organelle are absent in prokaryotic cell, it is not dividing into compartments. But in eukaryotic cell, see now there is a cell that have mitochondria, chloroplast, endoplasmic reticulum, Golgi complex, lysosome, vacuole, everyone is a what? Compartment. Got the idea? So because these membrane bounded organelle are absent in prokaryotic cell, hence we are writing no compartmentalization is present in prokaryotic cell. Are you able to understand? Cytoplasmic streaming means, okay, if you ask, I am saying cytoplasm of a eukaryotic cell always rotating in clockwise direction. It is in dynamic state, it is not static, it is always rotating in clockwise direction. It is called cytoplasmic streaming. You got the idea what is cytoplasmic streaming? Cytoplasm of the eukaryotic cell, inside the cell, when you observe through a microscope, you will find cytoplasm is always rotating, always rotating. It is called cytoplasmic streaming. That type of rotation of the cytoplasm not occur in eukaryotic cell. Picture came? Done? Clear? Compartmentalization I told ki in eukaryotic cell, inside the cell, many cell organelle are there. Sorry. Many cell organelle are there. So these every cell organelle making a compartment. So this is a mitochondria. It is a compartment of mitochondria. Another, suppose that it is chloroplast. So it is compartment of chloroplast. Every cell organelle behave as a compartment. So these compartment is present in which one? Eukaryotic cell, because membrane bounded organelles are there. In prokaryotic cell, these membrane bounded organelles are not there. Hence we are saying compartmentalization is absent. Hence we are saying what? Compartmentalization is absent. I hope you understand. Clear? Clockwise direction means what are the direction of the wall clock? You see na? It is my wristwatch. So what are the needle of the watch moving the direction? It is called what? Clockwise direction. Clear? Done? Is it all right? So one student asking, not understood, sir. What beta you not understood? Tell me, I will explain, don't worry. Which point you not understand? One more time about compartmentalization. Okay, look, I am drawing a prokaryotic cell. This is cell membrane, assume, and I am drawing a eukaryotic cell membrane. So in eukaryotic cell, nucleus is there. Mitochondria is there. Suppose that chloroplast is there. Suppose that endoplasmic reticulum is there. Suppose that Golgi body is there. So you see this, it is a eukaryotic cell. This is divided into many compartments, 1 compartment, 2 compartment, 3 compartment, 4 compartment, 5 compartment, likewise compartment, chamber is there or not there. Every cell organelle making a compartment. Here these all are absent. So it is prokaryotic cells. Compartment is there, not there. Compartment is there, not there. So many cell organelle are making a compartment. That's the reason I wrote who is absent. Compartmentalization is absent. So why cytoplasm is streaming, I will say about the answer later on. Got the idea? It required to teach cytoplasmic streaming, at least we talk at 15-20 minutes on that. Getting the things or not? So I told we will, why it is moving in clockwise direction, why it is moving, how it is moving. There are so many things in your mind. Don't worry, we will talk about that particular area later on. Got the idea? What is RNA? It is a biomolecule. I will tell about what is DNA, RNA. See, first thing I am telling you how to understand the things and learn the things. See, you are very new student for a particular subject. You studied biology in 9th, 10th class. Many terminology are coming. I also know it is coming. I wrote cytoskeleton. You don't know what is cytoskeleton. You want to know about it. I also know it. You don't know about cytoplasmic streaming and what it is role. You want to know about it. You are not aware much about compartmentalization. So you also want to know about it. Actually, what happened? If we going to fix every terminology detailings at same place as I wrote, it is not possible for us. So it requires, I am going to teach these difference between prokaryotic and eukaryote at least 20 days. Only we are teaching what? Prokaryote and eukaryote, because we are going to detailing of every word at the same spot. What is the best way during a study? What we need, what I personally as a teacher feel, this word is required to explain at this juncture. I am going to explain that work only. And at that point only, if we feel no, we will deal it later on the coming days. Because you see your NCERT, their ribosome is a cell organelle. So about ribosome 70S, 80S, we will teach at that point. Got the idea? Are you getting the idea or not? What I want to convey? So otherwise what happened? These difference I am going to teach, suppose that 20 minutes about cytoplasmic streaming, 30 minutes about cytoskeleton. Got the idea? So what happened? You also get field bore, sir is dealing only the difference between prokaryotic and eukaryotic cell. How many? 10 days, 15 days. So I will justify every line. I know where what to deal, but at the right time. You got the answer of your question. Question is good. Curiosity is good. Keep it up. But at a specific time, we will talk about it. Got the idea? You know what is cytoplasmic streaming, why it is moving in clockwise direction, why it is there, why not there in prokaryotic cell. I can understand it. These are some of the questions, of course, came in your mind, but there is a right time to answer all these questions. Got the idea? So don't worry about your every problem will fix, but a specific time in a specific chapter. Is it clear? Okay, ji, can we move ahead? Now we are moving ahead. So these are some of the difference between prokaryotic and eukaryotic cell we have seen. Now I am going to deal in detail about prokaryotic cell. Here you will got answer of many of you question asked. You ask na ki sir, how it will do the respiration, how it will do the photosynthesis, where energy will produce. So now I am going to teach you in detail about prokaryotic cell, and many of your questions answer going to satisfied here in prokaryotic cells. So where these prokaryotic cells are present? So example of organism where prokaryotic cells are present. So you can say prokaryotic cells are present in cyanobacteria. These all are a type of bacteria. I will tell later in a separate chapter. Eubacteria. A question came in your mind now. What is cyanobacteria? What is eubacteria? Getting the things or not? Eubacteria. Archaebacteria. There is a separate chapter where we will talk about all these type of bacteria. What is eubacteria? What is cyanobacteria? What is archaebacteria? Getting the things or not? Mycoplasma, actinomycetes, etc. So these are the some of the organism like what? Cyanobacteria, eubacteria, archaebacteria, mycoplasma, actinomycetes. These are some of the organism who is present. Prokaryotic cells are present. Got the idea? One student asking, sir, methanogens is also prokaryote. Answer is yes. Methanogen is an example of archaebacteria. Methanogen is an example of archaebacteria. That I will teach you in a separate chapter about all these in a chapter that is called biological classification. In this chapter, we will talk about details of it. What is archaebacteria? What is eukaryotic? What is cyanobacteria? What is mycoplasma? What is actinomycetes? How they differ from each other? What are the example of it? Got the idea? Is it clear in the mind? Is it all right? Right. So these are the some of the prokaryotic cells. Is it clear in the mind? So usually prokaryotic cell are smaller in size. Smaller in size. An example I am writing, mycoplasma. What is the typical size of a mycoplasma? Anybody can tell me? I am telling about size of a typical bacteria. These are the prokaryotic cell I wrote. I am writing some eukaryotic cell. Can you tell me the size of RBC? RBC, red blood corpuscles, is a eukaryotic cell. Here it is prokaryotic cell. Can you tell me the size of ostrich egg? Can you tell me the size of nerve cell? So here these are the some of the example of eukaryotic cell I have taken. I am trying to justify it, who is bigger, eukaryotic cell is bigger or prokaryotic cell is bigger. So tell me the size of mycoplasma now. It is in your NCERT. I just want to check how many of you are reading. Mycoplasma. What is the size? Too many differences are coming, answer are coming. Open your NCERT and tell me the size of mycoplasma as well as tell me the size of PPLO. PPLO is an example of mycoplasma. This PPLO is what? Example of mycoplasma. So what is the size of PPLO written in NCERT? What is the size of mycoplasma written in NCERT? So I am asking one by one. Otherwise, I am not able to connect with you whose size you are writing. Wait a minute. So start with PPLO. Now start. PPLO size. 0.1 mm. Na ji na. Mm means which class NCERT? One student asking. First year. 11th class NCERT. Mm means what? 0.1 micrometer. 0.1 micrometer. You are saying the size for which one? PPLO. Very good. Size of mycoplasma, what written in NCERT? Size of mycoplasma. .3 micrometer. What is the size of a typical bacteria written in NCERT? What is the size of a typical bacteria written in NCERT? 3 to 5 micrometer. What is the size of RBC written in NCERT? RBC 7 what? Micrometer. Now what is the size of ostrich egg is written in NCERT? Not written. It is largest cell. Largest cell is ostrich egg and nerve cell is longest cell. Longest cell. If you talk about ostrich egg, I am writing the size. It is in centimeter, 15 centimeter long and 13 centimeter wide. 13 centimeter in wide. It is in centimeter. And it is longest cell. It may be centimeter to meter in the length. Got the idea? Understand the idea? So PPLO is the smallest cell known. A smallest cell known. Got the idea? Now you just try to understand. When you read these data, you find fluctuation in the data. There is a reason. I am asking a funny question. Try to understand. Try to understand. I am asking what is the size of a mammal. You can ask what nonsense question you are asking, sir. I am asking what is the size of a mammal. Sir, RBC does not contain nucleus. Here I will answer it. I will answer it. I told dear children, what the right time for a particular question, I will answer at that time only. Got the idea? So it is not possible to fix every related question at a same juncture. Got the idea? That is a question we will deal in physiology. Any cells, one student asking, sir, why is ostrich egg a cell? Because ostrich egg perform all the function of their survival. Got the idea? So come on my question. If I ask a question, what is the typical size of a mammals? You can say, sir, it is a funny question. How can I say? Mammals is rat also, mammal is elephant also, mammal is blue whale also. Whose size you are asking? So if I wrote a size of a typical bacteria, thousands of different bacteria are there, all have a different size. So typical bacteria means we just put in an average. Okay, these are the average size of a bacteria. Are you getting the idea or not? It is averaging we did. It is not like every bacteria is 3 to 5 micrometer. Getting the idea or not? Is it clear? Understand? Is it all right? Right now? So if we talk about typical, I will show you the first slide in the last. Wait for that. Because we are in continuity of teaching, na? And if it is their group with you, definitely I will ask them to share the screen also. Getting the things, right? Haji. Now we are moving ahead. So what the conclusion we achieved? Who is smaller? Prokaryotic cell. Based on this data, prokaryotic cell is smaller in size, is smaller in size than eukaryotic cell. Eukaryotic cell. But one thing is there, prokaryotic cell divide faster than eukaryotic cell. Who divide faster? Prokaryotic cell divide faster than eukaryotic cell. Now we are coming on the shape of the cells. Shape of the cell. Size you have seen. Shape of the cell we are going to discuss. So different cells have variety of different variety of shape. Different variety of shape. It may be round, oval, spherical, cuboidal, columnar, right? Cuboidal, columnar, polygonal, irregular. So many different variety of shape a cell have. Got the idea? So what kind of shape a cell have? It's depend upon function performed by the cell. Means what are the function of the cell that decide the shape of the cell? Function performed by the cell. I am trying to make you understand here. Suppose that a cell is there whose shape is like this. Cell membrane is like this. So this is your blood vessels. So here you are finding the many enfolding present in the cell. These are what? Basically increasing the surface area for the absorption. So any cell who are increasing the surface area of absorption of material, whose purpose to absorb the material in large amount, you will find that kind of enfolding are present in the cell. I have given you an example how cell structure depend upon the function performed by it. If a cell is there who have many enfolded cell membrane, many enfolding present in the cell membrane. This is for which purpose? Increasing the surface area of the absorption. Are you getting the idea or not? So, moral of the story is I want to convey one thing to you, ki what are the structure of a cell? It depend upon what kind of function it is going to perform. If we talk about human RBC, it is passing through small capillary. So it is which type of a structure? It is a biconcave structure. If you talk about human RBC, it is which kind of a structure? It is biconcave in a structure. So these biconcave structure easily pass through which one? A small capillary. Got the idea? Understanding the idea? Is it clear? So shape of the cell depend upon what basically? What are the function performed by the cell? So which cell help to perform a specific function, that is going to decide the which one? Shape of the cell. If you talk about absorption, so many brush border are there, microvilli is there, enfoldings are there, they are increasing the absorption surface area. Got the idea? If it is somehow a small capillary through it travel, no resistance required, then what happened? It have somehow disc shape and biconcave type of cells. Understand the idea? Clarity came in the mind? Is it all right? Which one should I explain, dear? Which one? Which point you want to explain again? Is it done? Clear? One student asking, sir, I am from state board, so I don't know about NCERT. Okay, let me tell you about it. See, either from whichever of the board you are there, if you want to become a doctor, so for you, it is very important to read NCERT of all physics, chemistry and biology. Got the idea? So first you please present. Purchase this NCERT or you download the NCERT. Got the idea? So see, one student asking ki what the changes, I got your problem. What changes problem will arise when shape of RBC not driven by concave? In a disease, RBC shape change to a sickle shape. It become like a sickle shape. Got the idea? So that is called sickle cell anemia. What happened? These sickle cell RBC trap in the blood vessels and it causes hemorrhage. Blood vessels may rupture in any part of the body. Are you getting the idea? Even its oxygen carrying capacity decrease. So every relevant answer we give at a relevant time. Otherwise what happen? A big mess will create with everything. So these are another topic where you will study what happen when the shape will change. What problem will occur, what disease will occur, what symptom of disease will come, why the shape change. Everything we will study, but in a separate chapter. Got the idea? For everything there is a explanation is there, but please wait for the right time. Definitely I will satisfy your every question, every single question, but at a particular specific time will come for that. There is a separate chapter. You will study not in first year, you will study the answer of this question in second year. What problem will arise, sir, when its shape will change? Why shape will change? What is the reason behind change in the shape? What happened? If shape changes occur, which problem occur, which disease occur? You will study in second year. So it is not the right time. That is the reason I told you people 3 times today for answer a specific question related to a particular term. There is a special time is there. We will work on that time. Got the idea? Is it clear in the mind? So don't worry, you don't think like sir not answering my question. There is a reason sir not answering right now these questions because it is not related to here. Otherwise we can't move a single page in NCERT. Even 10-20 days I teach, I not move a single page. Got my point? Are you getting the things? So everything we will work out. Don't worry about. So NEET exam syllabus, first you keep in mind it is NCERT. Anything more than NCERT, definitely when you will come, we will say. Meanwhile, you think like 99% of the NEET syllabus is covered when you are reading the NCERT. Got there, children? Clarity came, right? Are these prokaryotic cell are microscopic in nature? Yes. Anything less than 100 micrometer, not visible by our naked eye. Got the idea? You just buy the NCERT book or you download the NCERT book from the app. E-Pathshala is the name of app. If you are not accessible, best is you purchase the book. It is not expensive book. And you just download this app. You go for 11th class. You go for biology. In app, you go for English medium. So you follow the path after downloading the app. NCERT will come in your mobile. It will come in your laptop, wherever you want. Through Play Store, you have to download it. Best is what? Purchase. If any new book shop is near you, you can purchase it. NCERT not provided by college. Study material of the college definitely we will provide. It is 100, 200 rupees book. You can purchase it. Right? Okay. So you will got it. You go through the app, you will got it. Okay, dear children, bye. So see you in the next class. In next class, we will talk more about prokaryotic cell and their answer of your many question will solve. Ki, sir, if mitochondria is not there, then how you do the respiration? If chloroplast is not there, then how do it photosynthesis? So what many question you asked today, their answer automatically you will got after finishing of the next class. Okay, ji? Done? Clear? So have a good day to all of you. I hope you understand the difference between prokaryotic and eukaryotic cell. Got the idea? If you wish QR code, I am providing you it. All subject NCERT. Yeah, physics, chemistry, biology. You can purchase all subject NCERT of first year. This is the QR code. If you want to scan, you can. So if you scan this with your mobile, all the things I wrote on the board will come in your mobile. There is a link came. You can click it, but for a student, best thing is what? While I am writing, you also write. This is the best way of study. When teacher is writing, you also write. So what benefit you will got? In notes will come. You can revise it later. While writing, you just go through everything. Got the idea? You can buy on CRT by own. Yeah. So bye-bye, bye-bye, bye-bye. Thank you very much for your patience, for listening, and keep it up. Keep it asking. Definitely, every time it is not possible, you will just got immediate answer. But definitely, that things encourage us. Okay, our student thinking in very right direction. Bye. Have a great day. Bye-bye.